Air stripping of fish eggs, the effectiveness of the procedure and the impact on brood stock management in two salmonid fish, rainbow trout and brown trout.
Hi, my name is Radosław Kowalski. We are from the Institute of Animal Reproduction and Food Research, Polish Academy of Science in Olsztyn. We would like to present the air spawning method of salmonid fish.
Hand stripping is a traditional spawning procedure in fish. This procedure may cause damage to the eggs by hand, resulting in lower fertilization potential of the eggs. An alternative method utilizes gas pressure to deplete eggs from the fish body. This method is called air spawning. Air spawning is a method in which fish eggs are collected without massaging the abdomen of the fish. A syringe and needle are used for injection of the gas into the fish body cavity. Air stripping of fish eggs. In this procedure, gas pressure should be controlled, and the best condition is maintained when its value is below 1 bar. Airflow should also be controlled. For salmonid fish, 1.5 liters per minute is the efficient speed of gas exhaustion. An optimum speed should be maintained because higher speed may not obtain all the eggs, while a slower speed may unnecessarily prolong the procedure. The syringe should be connected with a screwed needle to avoid needle detachment caused by the gas pressure. The fish should be anesthetized and carefully dried out before spawning. The fish is placed on a slant bed, about 35 to 40 degrees. The needle should be quickly inserted below the abdominal fins. The eggs are freely removed by the gas until only singular eggs are being removed.